Hello, everybody. I'm Dr. Mingo Park, in charge of training for utilization of fumigation equipment and devices in Kolka program. In this time, I'm going to present the rearing and assessment of target insects for disinfection test. I'm a team manager of phytosanitary treatment in Pest Control Division in Animal and Plant Quarantine Agency. I made a presentation in following order: on introduction, rearing method of stored products insects, rearing method for agricultural insects, preparation insects, and mortality calculation. For the introduction, even in the same insect species, each insect species show different resistance to chemical pesticides depending on the development stage. Therefore, it is necessary to develop the disinfection method to the most resistant stage by conducting a disinfection test at each development stage. In order to obtain a successful test result, it is important to identify and secure the exact insect species identification and the growth uniformity of the experiment insects to be used in the test. The mass rearing technology for experimental insects should be provided for the disinfection tests, such as dose response tests or confirmation tests throughout the year. In this presentation, I presented the insect mass rearing technology and the experiment method in laboratory. The main pests in the plant quarantine as follows: mealybugs, scales, mostly in fruits, aphids, mice, thrips, whiteflies, mostly in vegetables, termites, ambrosia, bark beetles in timber, the mice, aphids, whiteflies, thrips, and corkflower. You can read later this one. Fumigants interact with the insect at low temperature. It is difficult to kill insects because the low respiration and increasing suction rate of fumigant and decreasing of gas volume. So that's why it's very hard to kill insect at low temperature. And the high temperature, the chemical reaction increase between fumigant and commodity. It could be increased phytotoxicity of commodity. Test insect should be quarantine insect would be the best if you have a special facility. Choose the most important dominant hard to control insect. After choosing insect tests susceptibility for each growth stage, pick the most resistant stage as a test insect stage. Remember, the stage you've chosen must live in target commodity. If resistance of fumigant is reported, choose resistant species. If you cannot test with the quarantine pest, you can use similar insect live in your country. But you have to check differences of susceptibility on quarantine and non-quarantine pests later. If you test with reared insects, breed well before experiment. The next one you can see later. Breeding container. Breeding containers, food and breeding species are needed for insect rearing. Acrylic cage are needed for breeding of agricultural pests. Containers such as glass bottles and petri dish and the plastic bin are required to store the products insect. And also need a round sieve. This one round sieve round sieve with holes to separate the insect from food. As food sources for pests, through the food pests are necessary brown rice, this one, and flour and yeast. Potatoes are needed as food of agriculture pests, such as mealybugs and potato tuber moths. Each host plants is required for food for aphids or mites. The breeding chamber or greenhouse are controlled temperature and humidity. Now we are moving to second topic, rearing method for stored products insects. Major stored product insects are rice weevil and red flower beetle and cigarette beetle and rice grain borer. These are often used as insects for fumigation experiments, and I will explain how to rear them. Not all of them, I just choose two of insects. These pictures show the eggs, larvae, pupae, and adults of rice weevil. The larvae are milk white colored with 2 millimeters of body size. Adults are brown colored with 3 to 5 millimeter long size. For the bleeding of the rice weevil, use brown rice and products with the rice germ. Fill a third of breeding container with brown rice and inoculate rice weevil in the container. Carefully apply the repellent to the entrance of container. The size of breeder is selected according to the number of individuals. The optimum temperature for breeding is 25 to 27. The relative humidity 50 to 60%. The larval weevil is developing rice by gnawing rice grain. So we should maintain temperature. We should maintain temperature and humidity well. It should not be. It should be kept dry. Next insect is red flower beetle, Tribulum castaneum, eggs are superior and milk white. The larvae and cylindrical, light yellow, and the head is yellow, this brown. The others are 3 to 4 millimeter long, reddish brown, and have a large antenna and club shaped. For the rearing case of red flower beetle, prepare a plastic container or glass bottle with a mesh lid. Red flower beetle feed is mixed with 95% wheat flour and 5% yeast. Based on 800 grams of food mix, 334 grams of wheat flour with wheat, 66 grams of yeast. Optimum temperature is 25 to 27 degrees Celsius, and the relative humidity is 50 to 70%. If the humidity is too high, the growth of first red flower beetles will be slowed down. Prepare 2 sizes of sieves are stacked and shaken. Food falls on the floor. The red flower beetle dung and eggs remains on the sieve underneath. They collect the remaining eggs and they put them in new container with food for rearing. This. Now I'm going to discuss a rearing method for agricultural insects. Major agricultural insects are Pranococcus cetri, Pranococcus longicornis, Pseudococcus crepus, Apis gospi, Mijus poscae, Tetranychus urticae, and so on. But I'm going to introduce the rearing method to a few insects of them. For the rearing Strus millibus, plastic containers with mesh or cages are required, and the sprout potatoes are required, like this. For food, pour water in the large tray, put the sprout potatoes in a small bottle or plastic container in a large tray filled with water. Inoculate larvae on the sprouted potatoes by a brush, like this, and put them in an acrylic cage for rearing. Dried potatoes, which have low moisture are better than potatoes with high moisture for breeding. Very rotten potatoes are removed and replaced with new potato. The optimum temperature is 20 to 30 degrees Celsius, and the relative humidity is 60 to 70%. If the humidity is too high, the potatoes will not causing a lot of fly larvae. To control humidity, put dried potatoes in cage. For breeding cotton aphid, an acrylic breeding cage with mesh is required. Cucumber and red pepper seedlings are required as host plants. Cucumbers, peppers, and paprika are used as food for cotton aphids after 20 days of planting. Put the cucumber and pepper seedling pots on the tray. The aphid infested leaves are placed on the new seedlings in an acrylic breeding cage. When the temperature is high in August and September, a natural navy, Apididus colomani, is generated in large quantities. Breeding temperature is 26 to 30 degrees Celsius. Humidity 50 to 60% is good. Natural animals lay eggs and parasitize inside the aphid's body. The natural enemy sucks the nutrients of cotton aphid and kill cotton aphid. It finally decreased the density of cotton aphid. Therefore, maintenance is particularly important in the early occurrence of the natural enemy. This one. The host of two spotted spider mites is bean seedling, this one. Soybeans, the hosts of spotted mite can be used as food for about 20 to 30 days after planting. The grown soybean seedlings are placed in a tray filled with water and inoculated the spotted mice on the hosts. If the leaves is too dry and withered, the nymphs gather the tip like this, tip of the leaf, and break away from the leaf. The new fresh leaves are provided every 2 days. Breeding temperature 25 to 30 degrees Celsius, humidity 50 to 60% is good. Diamond back moth can be reared with the young rape seed. In order to receive eggs, you need to make a spawning pony, this one. The spawning pony, this one. I put 65 grams of cabbage leaves in blender with the 500 milliliters of water. They put the crushed Chinese cabbage into a flask and autoclave at 120 degrees Celsius for 20 minutes. The clumped and flattened the aluminum foil. Pour the filtrated cabbage solution on the crumpled aluminum foil and sorry, and foil completely is synced in the wide bottle. Then after dry, the foil is cutted as small pieces. If we roll the cut spawning foil and put in the cage with the adults, they would lay eggs. The larvae, the hatch from the eggs, they immediately begin to feed the cabbage. Eggs hatch in about 3 to 4 days at room temperature and grow into second insect larvae in 5 to 6 days. The amount of food they eat increases from third to fourth instar. It usually takes 2 weeks from eggs to pupa. The breeding temperature is 25 to 26 degrees Celsius and humidity is 50 to 70%. If possible, place the cage in a well-lit place. Now, I'm going to discuss on preparation insects for bioassay. Rearing insects are used for fumigation experiments. At least 30 insects are required for the experiments. 1 replicant. Add some hosts on or diet in petri dish and place 30 pass in the petri dish. And the petri dish in this way, like this, in this way are put in the container or box for the fumigation experiment. These show larvae prepared for several repeat experiments and once investigated the mortality after treatment. The pests are used in field application experiments as follows. First, prepare 30 insects, 30 breeding insects in each petri dish. Insects are placed in large-scale field like the storage grain warehouse. It is ready to start the fumigation test when the petri dish containing insects stand like this. The last chapter is about the calculation of mortality. After the fumigation test, incubate them at a temperature of 25 degrees Celsius and the relative humidity of 60%. Adults, larvae, and nymphs are checked for the number of dead insects from 1 to 3 days after incubation. The hatching rate of eggs is investigated 7 to 10 days after opening. All experiments should be repeated at least 3 times. Mortality rate and adjust mortality rate is calculated with this formula. Thank you so much for listening my lecture. I hope it will be useful for you all. Thank you.